Can we get David Sinclair to the stage?
Alex, Jesus Christ. Okay, sit up. Thanks, Martin. Okay, we're on. Thank you, Vittorio, for the great presentation and the intro. I think we're all in the mood for some rejuvenation. It's late in the day. Nice to see lots of friends and old colleagues. And thank you to the organizers. You're always very generous and kind. And thank you for inviting me. Thank you all for coming. So we have a lot to talk about today. I'm going to give you an update. I was here last year, and we had just published the ICE paper on being able to test a large part of the information theory of aging, which I will get to. That paper came out in Cell in beginning of 2023. After 13 years of work, 46 researchers, 30-something labs, and 3 mortalities of our co-workers along the way. It was an incredible journey, and I was very proud to present that to you all a year ago. And we'd also published a little bit earlier than that we could also, not just in these mice drive aging forwards by altering the epigenome, but also, as Vittorio has told you, we were one of the lucky groups, I guess resting on Yamanaka and Belmonte labs, to be involved in the field of rejuvenation. And I want to give you an update largely on that work now. Where are we? I said that when I came back, I was going to tell you a number of things about the chemical reprogramming, about where the information lies that we think for rejuvenation, update on clinical trials. I'm going to do a lot of that today. So what is the information theory of aging? It's a combination of a lot of labs' work. I'm certainly not the only one to suggest that the epigenome is important. I became involved in studying the epigenome through fortune, good luck, working with Lenny Guarenti in the early and mid-1990s. On yeast aging, and many of you are young, so you may not remember those days. But what we discovered as a team in Lenny's lab, and I was a postdoc then, is that there's a set of genes called the sirtuins, named after the SIR2 gene, and SIR2 is a histone deacylase that controls the epigenome. SIR stands for silent information regulator, so information is key. And of course, because information was part of the genes I was studying, I've been fascinated throughout my career in what the heck is information doing as a subject in aging. And I became particularly fascinated with this as we learned in yeast and then in mammalian cells that we could see predictable changes, not just in one cell type, but in many different cell types and tissues and species, changes that were predictable in the epigenome. Epigenome used loosely, meaning histone changes, methylation changes that Vittorio has just talked about. We came up with a theory that's not called the information theory of aging, called more of a mouthful, the relocalization of chromatin modifiers theory. That was in 2008 with Philip Oberdorfer as a postdoc. And that really was the beginning of this theory. It was a predecessor. And what we showed in that theory, in that paper that was published, was that we could disrupt and accelerate epigenetic change in a way that mimicked aging by creating very precise non-mutagenic double-stranded breaks. And we could see that we could drive aging forwards in cells, and now we know even in animals to do that. And then with the later work by our lab and of course many others now, and Belmonte before that, and Vittorio was there, with that work, it was very clear that, to us at least, that there's a backup copy of youthful epigenetic information in every cell that we've looked at. I'll show you evidence for that, but I really do believe that that's true. It's, for me, the best explanation for how it's possible to have not just reproducible changes in the forwards of aging direction, but in the reversal as well. Somehow cells know which gene to change in reversal, by how much and in what direction. Where that information lies, we are working actively to figure that out. We think it's probably something of a nucleic acid material, but that's about all we are sure about so far. This is published, so if you want to read more about this theory, I'm not going to spend time educating about it today, but I will say that it's largely based, this idea, on the work of Claude Shannon, who discovered and formulated the mathematical theory of communication in the 1940s. And instead of a backup copy of information, which he used to build the beginnings of today's internet, we're talking about biology here, and we think that there's a backup copy that we can use through Yamanaka factors, or increasingly chemicals, to reset the cell, find the original information that's somehow laid down when we're very young in cells, and can even last decades. As I'll show you, we're reprogramming cancer cells. We think back to their original state in the body, and that cell line has been out of the body for 50 plus years. So 50 years is a long time to store epigenetic information. And as I mentioned, we think that cellular damage, such as double-stranded breaks, and now we know nerve crush, and others like Vadim Gladyshev have shown that even pregnancy is a stress on the body that can accelerate aging. There are a lot of things that cause these, what are now referred to as epimutations. And this is the RCM hypothesis, which I just talked about, the idea that the relocalization of chromatin modifiers, such as the sirtuins, and of course others like HDACs, are involved in this process, PRC2, Vittorio just mentioned. We can look at our data and also find very strong associations with the PRC2 complex, so it seems to be very key to this process and rejuvenation. So here's the summary of this talk. The update is that we're doing a lot of things. We've gone not just deep, but now we're going wide. We've almost got too much to study, and we're too excited. But what we're doing is, everybody's doing artificial intelligence, but what we're excited about is that we can find small molecules and screen them virtually and process those very quickly. We also do reprogramming of cancer cells, as I'll show you. We're doing a lot of epigenomics. Where is this backup copy? Trying to find out how that all works at the molecular level. Can we reverse cellular senescence? Right now, I think most people would say that's impossible. I can tell you that it's not. Is it safe? I'm not sure yet, but we're working on it. Lots of human organoids, because mice are not sufficient. And we do a lot of work on inflammation as well, which I'm not going to touch on today. I'm also not going to touch on the human clinical trial preparation. Jerry McLaughlin was here, and as CEO of Life Biosciences, a company that was spun partly out of my lab, he talked about that, and he presented great data on the beginnings of primate work to restore visual function to blinded primates. And hopefully next year, if all goes well, or if, maybe next year or the year after, we'll see. But human studies are definitely being lined up to test these technologies, first in the eye and then beyond. This is the system that we use in mice, monkeys, and hopefully in humans. It's an inducible system for safety reasons. We use a tamoxifen as the major inducer, and it works really well. And what we found is that we can turn on this system for a few weeks, sometimes for over a year in an animal, and it's very safe. The reason that we think it's particularly safe is in part because it's not getting to the susceptible cells as much, such as the gut lining, but also because we're not using c-Myc, which is an oncogene, and we think that's important. So we're using OS and K Yamanaka factors, not OSKM. And that's what we've been working with ever since we published. This was work that we did publish, so I won't go too much in detail, but to highlight something that you may not have thought about as much as me, and that is that the rejuvenation of an optic nerve or a retina and restoration of vision in an old mouse or a mouse with glaucoma is associated with not just changes to the DNA methylation, but gene expression changes that are uncannily similar to a young cell. And this shows this. These are genes that go up with OSK treatment, viral treatment, and down. And if there was no change, these dots, which represent each gene's expression, would be flatline. It'd be no change like this in the horizontal. And if it was accelerating aging, which obviously it isn't, it would be from the lower left to the top right. But what you're seeing is the opposite. You're seeing that, in general, genes that when the cells were young that were down that went up with aging go back down by pretty much the amount that they went, that they changed, and vice versa. So that's why one of the bits of evidence that I feel increasingly confident that there is a mark or a nucleic acid or something on or in or near the genome that allows these Yamanaka factors to reset really accurately, sometimes, as I mentioned, decades earlier. I mentioned senescence. So, we all stain senescent cells for beta-galactosidase, which you can see here. What you may not know is that using OSK and, as I'll show you, chemical cocktails that mimic these factors, we can pretty readily reverse a lot of the gene signatures of senescent cells and even get them to grow again in a healthy way. Now, we don't know in vivo if this is safe, but in vitro it's not that difficult. We have checked to see if they're oncogenic, are they expressing genes that might predict tumor genesis, and we don't see any of that. So we think that what we're doing is restoring the ability of cells to regain their identity, having lost their identity during aging. And this is largely the work of Jae-Hoon Yang, who's now a postdoc in Korea, as well as Chris Petty, who's here at the conference, and Thomas McDougall. And these are gene expression patterns that you can see, heat maps of 3 separate experiments, 3 different cell lines. I mentioned AI, and I'm inspired by all the work that Alex does. We're primitive compared to him, but what we've been able to do is use AI to our benefit. And one of the things we're doing, Thomas is doing, is to use AI to very rapidly detect young, middle, and old cells from humans or mice. And you can look at cells, or I can look at cells, I cannot tell the difference. A young cell looks like an old cell, it might grow a little slower. But you apply AI to the visual system and train it just for 10 minutes, and it can pick out a young, middle-aged, and old. It can even pick out how senescence was caused, or how many passages it's been through. And this is a very useful method now because we can screen not through dozens of chemical cocktails, but potentially millions of chemical cocktails. And it's really shown how we do it here. I'll just be brief. We're looking for genes in chemical libraries using robotics, and we take cells from different age people, 20s, 90s. We can make them senescent. And Thomas paints the cells. We used to use just one dye to look at nuclear localization, but now we use a variety of cell paints, and that gives us beautiful resolution. And what comes out the other end is very, very accurate. It's so easy now to tell if you've rejuvenated cells in high throughput. And we've now got chemical cocktails and even single chemicals that can rejuvenate cells, human cells, mouse cells, different cell types, that we're testing on organoids, tissues, and animals. We've applied it, for instance, to human brain organoids, little organoids that we grow in the dish. We can give them Alzheimer's mutations, and they develop signs of dementia in the dish. We can measure their electrical activity. We can rejuvenate that. We can get the electrical activity back again in those organoids using the genes and chemicals that we're applying, and even gene expression patterns go back in those little organoids to a more youthful state. So we're excited about testing these chemical cocktails as well in disease models such as the eye, and we're also going to be testing these on animals. This is an example of some of the data that Thomas has recently generated, where you can see a big difference in the ability to detect non-senescent versus senescent cells, the blue versus orange. But you can also see that it depends on how you cause cellular senescence. Most people just, or at least if you're not really into it, you just do beta-galactosidase staining and they're all blue. But these programs can tell the difference between how they got blue. And these are cells here that are treated with an epigenetic inhibitor, GSK126, which inhibits the PRC2 complex that Vittoria mentioned. That's an excellent model for aging, we think, epigenetic aging. And there are other types of senescence, such as doxorubicin, which creates genomic stress or genotoxic stress, and we can see the difference. So we've come a long way. I'll be able to tell you a little bit more about those chemicals in a minute. We've gone beyond the brain. Last time I was here, we were talking about cerebral organoids. Now we're doing hair organoids. We're looking at being able to accelerate the age of these organoids, because you might appreciate that because they come from human stem cells, they're age 0. They're very hard to make old. But we can use our ICE system, like we did with those mice, and make them genetically older. And we're looking to see if we can make the skin old and the hair old and then rejuvenate it. You can actually put these organoids in the back of a mouse and grow human hair. It's really quite grotesque, but interesting. And we're also doing inner ear as well. These are inner ear hair cells on the left. These are some of the organoids. This is work by Shen Tiao, who's now at the Bone Institute in San Diego. And he was overexpressing in a very neuronal specific manner in the hippocampus O, S, and K using viral delivery. As you can see here, he stained it in the hippocampus. And what he and now others have shown is that we can rejuvenate the brain and get the ability to learn again in old mice. And he also has, I think, unique data showing that he can recover learning and memory and even lost memories in Alzheimer's mice models. What I thought I'd point out that's interesting is some of the genes that are expressed. We're seeing changes in the EMT transition. So this is in the EMT transition that's involved in embryonic development, playing a role in rejuvenation, which is pretty interesting. But also what's exciting is some of these genes here are very long non-coding RNAs that may be involved, perhaps required, we're testing in this rejuvenation process. So RNAs, as Victoria has identified, is, I think, a really interesting area to explore. What about cancer? Now, we had a bet in my lab that reprogramming would make cancer worse or better. And you can judge yourself how you would vote. I was voting that based on the information theory of aging, rejuvenation would take any cell, whether it's old or cancerous or tumorigenic, and restore the identity of those cells. So my bet was on them returning back to normal. I didn't think it would work on every cancer cell line, but I'll show you an example of the results that Nalat in the lab, Siwapur and Chai, is doing. She's a graduate student, very talented one. This is the EMT data if you wanted to take a photo of the kind of processes that we see in the brain, get a snapshot of that. Yeah, so here's Nalat. Nalat has been working on this. I think she was skeptical too, which is always good. And what we know is that if we or others overexpress O, S, K, and M transcription factors in mice, it'll either kill them rapidly or create teratomas. So that's not going to be a therapy anytime soon. But we wondered whether if we leave out c-Myc, the oncogene, what happens to a tumor that's already pre-existing. And we're studying lung, kidney, colon, and melanoma. This is the data from colon cancer cells. This is the first cell line that Nalat worked on. I'm not showing this because it worked only. It's really the one that she studied first. And this is an inducible system. You can see it coming on the left. And these are the stains. So what happens after induction of OSK in this cell line? These, by the way, these come from the lining of the gut from over 50 years ago from a colon cancer patient. These are endothelial cells originally. This is a scratch assay looking at cell growth. If the cancer cells grow quickly, they close the gap in the scratch on the plate, and you'll see this graph go down quickly. And the only cell line that closes poorly is the one that's expressing OSK, which is shown in the green balls here. Similarly, hopefully you can see this up the back. The only cells that grow slowly in this video are the ones that are overexpressing OSK. All the other controls grow like weeds. So that's pretty cool, but what's happening? Are we just making them sick or what? Well, yes, they go through induced programmed cell death. And in studying them, what we're seeing are really interesting changes in the lysosome, in autophagy, and also their morphology. They are starting to resemble, or they at least are reminiscent of their original cell type. And one of the examples that I'm showing you here is the nuclei are long and extended, and you see these in the columnar endothelial cells that they originally were, again, 50 years ago. So that's a really interesting idea that, and we have to prove it, of course. I'm just showing you some of the early work, that reprogramming can restore the identity of cancer cells. And I think, this is again speculation, that when cancer cells wake up to their identity, they freak out and they kill themselves. That's what seems to be happening. We're doing a lot of epigenomics, trying to figure out how does reprogramming work. And this is just an example of some of the data that we're getting. This is by Sun, Mabry Lewis, and Chris Petty in the lab. And this is an example of where SOX2, KLF4, SIRT1, one of the sirtuins, and TET2 TER2 being a DNA demethylase enzyme, that they're going in large part to the same places during reprogramming. This is in the first 4 days of the process. Obviously, later is different. But the first 4 days, they seem to cooperate. And it's interesting that sirtuins are involved as well. And the kind of processes that they move to are genes involved in stress, DNA repair, and cell cycle. Again, really interesting. So just briefly about the small molecules. Remember, I was going to talk about that last year. It was just preliminary. We've come a long way. These are the chemicals that we published last year. And you can see them here. I think some people are already trying to take these. I wouldn't advise it. Some of these are not exactly known to be safe. But we're trying to make them safe. So instead of it costing $200,000 to rejuvenate a tissue, let's make it $200. That's our goal, and make it safe. The assay that we used to use, and we still think it's useful, is to look at whether proteins are leaking out of the nucleus, which happens during aging and senescence, or if they go back inside the nucleus. We call that rejuvenation. And it's a good surrogate for that. And this is a good example of the data that we get. These are leaky nuclei in the senescent cells, and after treatment with one of the cocktails called C6F, which is one of the more potent cocktails from last year, we get almost a complete restoration of nuclear integrity, even better than the gene therapy, which blows my mind. And this is largely the work of Chris Petty and Thomas again. So where are we at? We've done enough work on these chemicals that we've now identified sets of them, and even a single chemical that we're treating mice with. We know already that they're safe in vivo, and we're going for it. We're going to see if we can reverse senescence in vivo, cellular senescence, and hopefully lifespan will be improved, and perhaps health along with it. And that'll probably be for next year. But that's where we're at. So what I want to emphasize is that what seems to be apparent from work from other labs like Vittorio's and Juan Carlos Belmonte's lab, any others, Tom Rano, we are seeing that reprogramming can restore identity of cells from many years before, allowing them to behave like they were young again, quite literally because they are young again. And this is an example of that shown graphically where over time during aging, cells lose their identity, becoming this red ball. And Waddington would be proud. We now seem to be able to push the balls back up, back to where they came from, even if they're cancer cells in some cases. So I will end here, happy to take questions. And there's a lot of people I mentioned along the way. Yu-Chen Gao I didn't mention, I should have. He did a lot of the organoid work, and he's charging ahead trying to restore hair growth and gray hair. That'll be fun. And Minyan talks about inflammation, but I didn't have time to talk about her work today. And the many, many collaborators over the years. And I want to just point out, in particular, Vadim's helped a lot with clocks. Vera has helped out a lot with the RCM work, and John with transposon work. And John's here as well. So thank you very much. I want to thank the sponsors and for inviting me back again. Thank you.
Thank you so much. Thank you, David. We have questions.
Excellent presentation. And I have a question about small molecules. Do you think that your therapies can be eventually done with small molecules?
Oh, well, that's the goal. From what I've seen, I would say probably. I haven't seen, let's put it this way. I haven't seen anything that says that they're dangerous and that they don't work in human organoids or in cell culture. I don't know yet, really I don't know yet if they work in vivo, but I don't know why they wouldn't. Okay? We're going to be able to bathe all the cells in the body with these chemicals, which is different than gene therapy, which is very hard to deliver, and we'll see what happens. I think it should work. The question is, how safe is it? And that's obviously on our minds.
I will ask a quick question. There's a couple of questions in the back, but while the microphone is going there, then I will ask a question. So regarding the substance, the secret sauce, whatever it is that makes a cell identify as that particular cell, I was wondering, have you thought about like secondary DNA structures or something that's already in the genome that could affect DNA methylation in the area?
Yeah, we have thought about that. Yeah, we think there's some structures that are laid down like that that there's a whole different set of proteins that recognize those structures. I think it'll be similar to CRISPR, how that works. There's a barcode at the gene that tells it what it used to be, by how much to change it, and in what direction. Yeah.
Fascinating.
Hi, David. Very, very nice talk. Sorry, 2 quick questions. The first one is on the cellular senescence reprogramming. Are we talking like oncogene-induced senescence, because this involves complex chromatin rewirings? Or are we talking like replicative senescence, which I can see why that would be easier to reprogram?
We've reversed the 3 main types that I talked about. Replicative, chemical-induced, and genotoxic stress. Unless Chris is going to jump out and correct me, I don't believe we've tried oncogenic senescence, oncogene-caused senescence. But I agree with you that that may be harder to reverse.
Okay, thank you. And sorry, one more question.
One question, sorry, because we are running out of time, so we have to give more people chances here. Vittorio?
Nice talk, David.
Oh, hey.
Nice talk, lovely, thank you. I'm very excited about what you're seeing on the cancer cells, and for 2 reasons. Well, obviously, maybe one day we could think about partial reprogramming as an anti-cancer therapy, number 1. But number 2, I think also it answers the question that I keep being asked, that probably you also keep being asked, like what happens if by chance you target a pre-cancer cells as you do the rejuvenation, in the body. And I think it's kind of, well, new data needs to come, but we can even start thinking about the safety of the approach also as a way that, even if cancer cells get targeted, maybe there is a chance that actually those are going to go into apoptosis, die off, and so it's a higher degree of safety, I would say. Would you agree?
Yes, I agree. And that's one of the reasons we did the experiment, besides testing the theory, is that we want to know if it makes cancer cells more active, and we're getting the opposite result so far.
It's very exciting. Last question down there.
Yeah, thanks so much for the great talk. I was curious about the efficiency of the reprogramming with OSKM, because with OSKM reprogramming for iPSCs, some reports say that it's quite low, 1% or so, and I guess that varies, but I'm curious about with the partial reprogramming what the efficiency is and if there's ways to increase that.
It's really, really efficient. It's not like generating iPSCs. Would you agree? Where's Vittorio? Anyway, so when we did the retina with Bruce Cassandra's, and this was the Nature paper in 2020, we could stain for OSK expression in the retina, in the ganglial cells, in the neurons, and stain for the restoration of cell identity using a marker for RGCs. And every time the neuron got the OSK and it was expressed, that marker came on. So the delivery is the hard part. It's not the restoration of youth in the cells, which contrasts with iPSCs.
All right, thank you so much, David. We will move on. We have a panel after this, so really appreciate it. Thank you. We will have, let's have a 10-minute break. Come back at 8:30 for the pharma panel. It's going to be really exciting. And after that, we go to the bar.